Hello everybody, welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership GARDP. My name is Astrid Pence-Moore and I'm hosting this webinar on non-traditional antibacterial therapeutic options and challenges. For those of you who are joining our webinars for the first time, Revive is GARDP's education and outreach program, and it aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. Our webinars are part of our educational activities. All webinars are recorded in full and can be viewed after the live broadcast on our website revive.gardp.org/webinars. I encourage you all to visit the Revive website to stay up to date about our future webinars, watch the recordings of previous webinars, and also to find other information, such as our blog about AMR-related topics. As usual, today's presentation will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel, as shown on the slide. We will address the questions after our presentation, and we will do our best to respond to as many questions as possible. Today's speaker is Ursula Theuretzbacher from the Center for Anti-Infective Agents in Austria. For the Q&A, she will be joined by GARDP's Director of Scientific Affairs, Laura Piddock. The webinar will be moderated by Prabhavathi Fernandes, who is also the chair of GARDP's Scientific Advisory Committee. Welcome, Ursula, Laura, and Prabha. Prabha, I'm now handing over to you for a brief introduction of the speakers.
Thank you, Astrid. Welcome, everyone. I am happy to welcome also today's speakers. Dr. Ursula Theuretzbacher founded the Center of Anti-Infective Agents in Vienna, Austria in 1988. She's an expert on antibacterial drug research and development, what we refer to as R&D, with a focus on clinical and public health strategies and policies and their needs. A broad therapeutic area of expertise includes early integration of pharmacokinetic and pharmacodynamic concepts, commonly referred to as PK/PD, optimization of dosing and usage approaches, public funding strategies for antibacterial R&D, and initiatives to recover the global antibacterial pipelines. Previously, she was a work package leader or partner in several EU-funded international collaborative projects focused on antibacterial drug R&D and reviving of old antibiotics. Additionally, she served as president of the International Society for Antibacterial Infective Pharmacology. She was the founding president of the PK/PD of any infective study group of the European Society of Clinical Microbiology and Infectious Diseases, which is known as ESMID, and was an executive committee member of the International Society for Infectious Diseases, the ISID. Ursula was a member of the coordinating group of the WHO Project Priority Pathogen List for R&D, and is a leading scientist for the clinical and preclinical pipeline analysis and development of target product profiles at the WHO. She lectured at the University of Vienna for 10 years and was recently appointed the ECMID Fellow. She holds a PhD in microbiology from the University of Vienna and the University of Innsbruck in Austria. Welcome, Ursula. It is also my pleasure to welcome Dr. Laura Piddock, who will join Ursula in her presentation and the Q&A. Laura Piddock joined the Global Antibiotic Research and Development Partnership, or GARDP, as the head of the Scientific Affairs in January 2018 and is now the Director of Scientific Affairs. Laura is also Professor of Microbiology at the University of Birmingham in the United Kingdom. Her research focuses on multi-drug efflux pumps, transmission of drug resistance between bacteria, and developing tools for drug discovery. She has published over 250 internationally peer-reviewed journal articles, review articles, and has presented at many conferences and has published 6 chapters in academic books. Laura has advised organizations such as the WHO. Notably, the scientific data from her team has been used by national governmental agencies when deciding whether to withdraw the licenses of some antibiotics from veterinary medicine. Thank you for this, Laura. Until 2017, Laura was the British Society for Antimicrobial Chemotherapy's chair in public engagement, and in this role was the director of antibiotic action and led the secretariat of the UK All-Party Parliamentary Group on Antibiotics. She was also the chair of the EU Joint Programming Initiative on Antimicrobial Resistance Scientific Advisory Committee. Laura is a fellow of the American Academy of Microbiology and a founding fellow of the European Society of Clinical Microbiology and Infectious Diseases. It is with pleasure I welcome Laura and Ursula. Ursula, you can start your presentation now.
Yeah, thank you, Prabha, for this nice introduction and giving me the opportunity to talk about non-traditional antibacterial therapeutic options and also their challenges. A few years ago, we didn't know the term non-traditional. Since about 100 years, we are used to have a small molecule direct-acting antibiotics available that work very well. But now we have a lot of emerging technologies, a lot of different technologies, and we are summarizing them somehow as non-traditional antibacterial therapies. Since these 100 years of antibiotic discovery, resistance developed, and you all know the big problem of increasing resistance and the need for the discovery of new classes. That proved to be quite difficult, especially for gram-negative bacteria. And so somehow people said, let's try something else. In general, the evolution of therapeutic agents in all fields of medicine, from small molecules to therapeutic proteins to novel modalities, is visible. So small molecules are simple, defined structures. They are stable. They can be produced very well. The therapeutic proteins are large, complex structures, not so easy to reproduce, and come with a lot of challenges. Recently, I mean the last 10, 20 years, novel treatment modalities, especially in fields like cancer therapies, became visible, and this is a very diverse field of therapeutics. So this general evolution of therapies and increasing trend to something else than small molecules is also visible in the antibacterial pipeline. I researched this global preclinical pipeline to see what companies are working on. In this analysis of more than 400 preclinical projects from 314 institutions, I found only less than half of projects focusing on direct-acting small molecules, so the traditional antibiotics. All the others were non-traditional therapeutic strategies, very diverse, from potentiators, just repurposing something that's already available, antibodies, vaccines, immunomodulators, anti-virulence approaches, phages, and microbiome-modulating therapies. When looking at the clinical pipeline, so what's really going on now in clinical development, it looks very different. Not many therapies have made it to clinical development already. You can see here it's mainly antibodies. We know them already, and some are also in clinical use, live biotherapeutics, I will come back to all these strategies soon. What is really important to know is if these strategies, if these therapies are direct acting, that means are they inhibiting or killing bacteria directly or not? As you can see on this slide, not many of these strategies are following this principle of direct acting strategies. It's mainly phages and endolysins, sometimes also live biotherapeutics and antibody conjugates. I will now focus on a few of these more common strategies. And these are antibodies, microbiome-modulating therapies, and anti-virulence strategies. A few words about anti-virulence approaches. This is a very diverse field. This could be antibodies, could be small molecules, could be nanoparticles. They could be used for prevention or for adjunctive therapies. Remember, they are indirect acting and therefore can only be used for adjunctive therapy together with an active antibiotic. In general, the main targets of these antivirulence approaches are Clostridioides difficile, Staphylococcus aureus, Pseudomonas aeruginosa. But of course, all other antibiotics can also be targets. Virulence factors are very diverse. There are so many described, and you find a huge body of literature about virulence factors. So I chose some examples here, which are the target for some clinical projects. Exotoxins are very logical target, especially if they are responsible for the symptoms of a disease. C. difficile is very well known with its toxin production. So these toxins are exotoxins and responsible for symptoms. But also other pathogens have exotoxins that are targets for these approaches. Virulence factors are secreted, and especially in Pseudomonas, there is a very well-known secretion system. It's like a syringe-like secretion machine with associated proteins for regulation and translocation. All that is essential for pathogenicity, but not for viability of Pseudomonas. This anti-PCRV, this part here of this syringe is the target of antibodies and inhibits toxin translocation. Communication, called quorum sensing, is also a long known principle, very important, and is responsible for signaling and regulating a lot of different processes. Immune invasion is important, especially also for Staphylococcus and also Pseudomonas. The regulation of virulence factors is a very broad approaches and known for Staphylococcus. So when we are now looking at challenges for these approaches, but please remember they are very diverse, and each of these approaches has their own set of challenges. But in general, selection of target. There are so many virulence factors, and usually you cannot really target all of them, so you have to select. And if you just look at Staph aureus, how many of these virulence factors are available as targets, you can imagine how important it is to target the right one. These virulence factors are not expressed uniformly in all strains, and they are also not always expressed. So there are differences in geographical situations, and choosing the right one is very important. Also, virulence approaches may be effective only in specific disease states, like chronic disease or dormant populations, or at specific time point in the infectious process, or at a specific infection site, or in specific patient groups. This is a challenge if it comes to selection of patients and planning clinical trials. Diagnostics, routine diagnostics, are usually not sufficient for antivirulence approaches. So diagnostics may have to be developed specific for a special situation. Dose finding is not easy because predictive models to support dose finding are mostly lacking. The same is true for predictive models for efficacy. In other direct inhibiting direct working drugs, we have models that are really very well predictive for clinical efficacy and also for dose finding. This may not be the case here in antivirulence approaches, and we still lack a lot of studies and models that would help with the development. So far, it's more a search and fail strategy, and it needs a lot more work to be done. It's also very important to know if this approach has a clinically relevant impact on the disease, because otherwise the clinical development will not be successful if the outcome is not clinically relevant. And that brings me to a very general problem of clinical development, that superiority studies are essential to show that adjunctive therapies provide a benefit to patients. And superiority studies are quite difficult to do. The next field is nanoparticles. Nanoparticles are not new, but are now increasingly studied. And I've noticed that especially in academic settings, this is a very popular field right now. As I said, it's not new. We know nanoparticle-coated devices. We know nanoparticles as carriers. We have had that in the past. They are loaded with antibiotics or antifungals to protect against deactivation or to improve efficacy or improve safety in pharmacokinetics. Nanoparticles can also have intrinsic antibacterial properties. For instance, they could also trap toxins or work directly. Technically, nanoparticles could be metal oxide or nano combinations, or carbon nanotubes. So these have their own challenges, especially if they are applied systemically and not topically. They have also specific hurdles for affordable large-scale manufacturing. Yeah, the next big field is the microbiome. Microbiome maybe you remember many years ago, we would say we need to eat yogurt to support our normal flora. And then we introduced this new word of microbiome. And since this time, it's much more interesting. But of course, it's not just the name, it's mainly the new technologies that are now available to study such a complex issue like the microbiome. Without these new technologies, we wouldn't be there. But still, we don't know much, at least not enough to really understand the implications of changes of the microbiome or what to do against it. So it's incredibly complex with about 5,000 different species. But what is so important is not just the number of different species, but how they interact and how these disturbances of this complex microbiological society may manifest in discomfort and different illnesses. So we still don't know much, but still our understanding has greatly improved, mainly due to culture independent approaches and also computational advances in the metagenomic field. So advancing science is really great. Because of this new knowledge, the microbiome market increases, investment increases, because so many indications are now affected. And sometimes I think everything is now attributed on to the microbiome. But we will see what or how this field will develop. We can for sure see that the number of publications and patents increased dramatically in the last years. So the microbiome-modulating therapies are mostly indirect-acting therapies that could be very complex or very little complex. And I think that's a real important fact. So the most complex therapy is fecal microbiome transplant. That means that the total complex microbiome is transferred from a healthy person to a sick person. It's mainly Clostridium difficile infection. And this strategy works quite well. Yeah, you transfer complex things in their whole complexity to a patient. But now the trend is going to less complexity. For instance, selecting natural microbiota components and have more control over the therapy. It could also be even less complex to selecting or deciding to engineer bacteria or phages to do very specific things. And this is especially because we have these CRISPR-based technologies available now since a few years. And more ways are explored to use these technologies. Examples are CRISPR-engineered bacteria to influence the microbiome, CRISPR-engineered lytic phages to eliminate pathogens, or CRISPR-engineered temperate phages to control gene expression and thus modulate activity of bacteria. As mentioned already, there are also direct-acting therapies that deliver antibacterial payloads and so work directly on specific components of the microbiome. These are usually engineered phages or engineered bacteria. If we look at the preclinical pipeline, what is researched? What are the projects out there globally? Then it's going in the direction of engineering strategies, engineered bacteria. So in former times we called these probiotics, but they are engineered to fulfill a very specific task. Or it's in a selected natural microbiota strains. There are diverse other projects like delivered payloads by phages or antibacterial compounds against specific bacteria. So there are a lot of different approaches that are used to modulate the microbiome. So what are the trends in microbiome modulating therapies? It's in general life therapeutic products. That means everything that's involving living microorganisms that are usually engineered to have very specific characteristics. In general, it means reducing ecological complexity, but at the same time, increasing technological complexity. An example for this is using phages as vehicles. So you have then two main challenges. One is the phage as a vehicle, and the other is what it's carrying to your microbiome. So a lot of technological complexity. Yeah, there are still a lot of challenges because despite such an increase in knowledge, we still lack a lot of insights into really basic problems of microbiome modulating therapies. We have an incomplete understanding of the complex genomic and phylogenetic diversity. We just know it's very complex and have mainly statistical and artificial intelligence strategies, but who knows if that's really enough to understand this complexity. The reduction of this complex ecological system may not translate into medical meaningful benefit. So it's very difficult to to understand the translation from the concept to the clinical effect. We usually have a lack of standardized, validated, predictive preclinical models. The dose finding is also a problem. Just knowing if live microorganisms are really integrated then in the microbiome and stay there and all these things is really difficult. Manufacturing of live therapeutic products, all this quality issue in manufacturing is not trivial. Also the intended use. So what is the best intended use beyond Clostridium difficile infection, where we know most, but the trend is going towards other applications. So where is the best medical benefit? And that leads to the translational challenges, the clinical development, if it's preventive or adjunctive approaches. So a lot of challenges, scientifically very interesting, but we have to translate these concepts into clinical benefit. With this, I'm coming to the next big topic, that's phages and phage-derived agents. Phage therapy uses a natural phage lytic cycle with the release of new virions at the last step and subsequent infection of new cells. The other option is endolysins. Endolysins are enzymes that are produced by phages and hydrolyze the bacterial cell wall. And the cell wall bursts, as you can see here. So I'm coming back to phages. The most common trend right now is the trend toward patient-specific phage cocktails based on established libraries of pre-approved phages. So a lot of phages are isolated, they are characterized and put into a phage library. If there is a patient with refractory infection, no antibiotic works, the bacteria are isolated and screened for their susceptibility to phages in this phage library. If the right phage has been found, then the patient is treated, either cured, or if resistance occurred, then a new phage is searched for in the library. So this is a very common trend right now to do. But also there are a lot of other strategies. Advances in molecular biology and synthetic biology have enabled this rapid progress of phage engineering. And scientists try to optimize natural phages and to address some of the problems. I just want to highlight this expanding host range and host range switching of phages. So which phages are, phage therapies are in preclinical development? It's still natural phage cocktails, but especially engineered phage cocktails, but also engineered endolysin, and I will come back to this a little bit later. Phages are addressing very different bacteria. It's mostly Pseudomonas, but also Staph aureus and many different other species that are addressed. So the major challenges of phage therapy is host range. You need to find phages or phage cocktails that have a really broad host range. Otherwise, it gets really difficult to find the right patients for the cocktail. Yeah, CMC, so quality manufacturing, especially production, stability, purity, these are all challenges. But I've seen really also advances in this field recently. Pharmacokinetics, this is a huge issue that has not been really solved yet. Distribution, dilution in the blood compartment, rapid clearance, and kinetics of phage infection, dependence on bacterial load, exposure and efficacy relationships. And these are all the reasons why so far mainly topical administration has been used. Also immune response, that may be possible. if you want to develop such a phage therapy, then selection of the infection. So not every infection would be suitable and is phage accessible. Showing efficacy in clinical trials is a huge issue. Developing and implementing appropriate diagnostics is also a major challenge. And this is all really shown by many recent failures of beginning clinical development, and we are not that advanced yet with phage therapies. So to summarize these challenges, the success of phage therapy ultimately depends on the optimal dose, dosing regimen, timing, formulation, and administration, with PK and PD, so pharmacokinetics and pharmacodynamics, characterized for each phage or phage cocktail. These are really important things. I mentioned already endolysins. These are the phage-derived enzymes. Also, lysins are well known. They have been now revived, and clinical studies were also described in the 1960s and 1970s. But now this revival is impressive right now. So endolysins are phage-derived enzymes with huge structural and biochemical diversity. They degrade the bacterial cell wall here, the peptidoglycan, in gram-positive cell walls. But recently we have also seen projects that tried to find lysins against gram-negative bacteria. For this, the major hurdle is the outer membrane of gram-negatives and engineered endolysins that enable the penetration to the peptidoglycan here have been developed. I'm coming almost to the end and just wanted to mention some recent strategies. They are also not new, but we see them right now in some preclinical projects. And these are indirect acting approaches on a population level. So the goal of these approaches is to achieve a population level benefit, public health benefits. There's no or little benefit for individual patients, at least not immediate benefits for an individual patient. So they may either prevent colonization or reduce transmission or prevent acquisition and spread of resistance determinants, or they may reduce the resistance reservoir in the microbiome. So on a scientific level, it sounds very interesting and intriguing, and the potential public health benefits are assumed, but not proven. And as long as you cannot prove something, it's really difficult. So the challenges for these approaches are that we don't have surrogate markers that may predict clinical benefit. The clinical development to get approval currently is not possible, and there wouldn't be a market acceptance. So as long as you cannot show a benefit to the individual patient or even to the population, it will probably not work. And there's this EMA guidance that says indications that relate to the reduction or eradication of a pathogen from a specified body site are not acceptable unless the microbiological effect of active treatment has been shown to result in a measurable clinical benefit. So even if they are really interesting, these approaches and should be studied in general, it doesn't seem to be possible at the moment to develop such a drug because you cannot show that it works. So this is independent of regulatory agencies. You have also to show the user that it works. So with this, I'm coming almost to an end. The clinical development of non-traditional therapies in general is a challenge because you have to select an indication with sufficient patients for clinical trials. That means it has to be common enough that there are cases that you can include in a clinical study. You have to show a meaningful benefit for patients to justify its clinical use. That's again independent of the regulatory agencies. Without showing a benefit to patients, it's not very useful to have such a medicine. Validated meaningful surrogate endpoints are often not available, and much more research, much more relevant basic and general research needs to be done. And then, of course, phase 3 clinical trial programs to achieve approval are also necessary. And here are coming again these two principles that have direct implications for the clinical development. These are the direct-acting drugs. These are our traditional antibiotics. These are standalone bactericidal or bacteriostatic drugs, and they can be studied in non-inferiority, could also be if needed superiority design for phase 3 trials versus standard of care. So you can choose whatever indication and show non-inferiority against standard of care. It may not convince payers to pay a lot for non-inferiority, but it's a starting point. You can get a drug approved. For indirect acting drugs that are usually adjuvants, because they still need an active antibiotic, you have to combine them with standard of care. But showing superiority against a well-working antibiotic and showing that's even better than this well-working standard of care is really challenging. It needs to be discussed to find some indications that would make it possible to show superiority. For prevention, you also have to show superiority against placebo to be able to show that this preventative approach really makes a difference. So if you would ask me, what does it mean for the future? Then it's really hard to know what's happening. For sure, we are advancing, we are really making progress, and new technologies will probably help us to advance, like it happens with medicine and therapeutics all the time. We are advancing. So maybe one of these non-traditional therapies will be in the future a great addition to what we have right now, especially in times with these increases and resistance, but also with still indications where we could really need an improvement over the currently used antibiotics. So the future is open, science is interesting, and I really hope that we can see some successes in the future. So I hope I could give you some overview of this field of non-traditional therapies, and I'm handing over to Prabha.
So thank you very much, Ursula, for your very thought-provoking presentation. We can now start the Q&A session. As a reminder for the audience, you can submit your questions as shown on the slide. We will do our best to respond to as many questions as possible during the allotted time. We are planning to finish at 12:30. So let us start. We see a couple of questions here. So the first question, let me address that for Ursula. It's from Maro Gote. Isn't there the same problem of developing resistance for phages as we have for antibiotics, or is there a difference? This is a very important question, so let us talk about that, Ursula.
Yes, this is clearly a very important question, and there is indeed a difference. For phages, developing of resistance is just a normal case, and development of resistance is very fast, and could be just the next day of therapy. And that's the reason why you can usually, you should not or cannot really use a single phage therapy, even if it works in the beginning. This is the reason why you usually have phage cocktails or also phage libraries where you can adapt the cocktail and include new phages and eliminate the resistant ones. So this is a real a challenge for phage therapy, but it can be managed with phage cocktails and patient-oriented therapy, and really adapt the therapy during the course of the treatment. With antibiotics, luckily, it doesn't happen that quickly.
Thank you very much. Here's another question. It's from Timothy Schultz-Ramo. Thanks, Ursula. What are your thoughts on RNAi gene silencing as a non-traditional approach option if they can efficiently be delivered into gram-negatives? I think that's a big if, but Ursula, it's all yours.
Yes, I mean, it really depends exactly what strategy it is. What kind of what RNA it is that you are silencing, is if it's something that would kill the bacteria, or if it's something that is silencing a resistance gene. So in general, this is a non-traditional approach, but really depends on the strategy. So I guess that Laura would like to add something here.
Laura, you want to take that?
Yes, so I think, as Ursula's just said, it's all about delivery, and the challenges of getting any compound, if it's small molecule or antisense RNA, is always getting it into the bacterial cell and getting it all the way through to where the target is. And of course, the target here is intracellular, and that's a significant challenge. So you almost certainly would need a delivery vehicle to make sure that you could transport the antisense in and get it to stay inside and be released at the site it needs to act.
Yeah, it's for sure there are two challenges. One is the delivery, so you need a vehicle, and then the real acting part.
Okay, I can maybe ask another question Laura, perhaps this is for you. Role of immunomodulator in treatment. Would you like that? Or maybe Ursula, but Laura, you want to try that?
So again, really, it depends on the patient, what type of patient they are and what sort of immune response they have that you're trying to modulate, the infection, i.e. the indication, and the pathogen. So it depends whether you're trying to stimulate the immune response or reduce the immune response, or whether you're actually adding an immune modulator. So this is actually a very difficult question to answer because context is everything. Ursula, I don't know if you wish to add anything.
Yeah, and also if the immune modulation effect is so essential that it really impacts the clinical course of the infection.
Very good. Let's try another one. Ursula, regarding indirect approaches, I understand the hurdle of demonstrating measurable clinical benefit in a population, but how about establishing global safety? And this is from Vid Stanilowick.
So I'm not sure if I understood correct.
Let me say it again.
Part, at least the last part, the second part.
Okay, so you can measure, let's say you can overcome the clinical benefit to the population. How
Yes.
about establishing safety?
Oh, I mean safety. You still give this drug or whatever it is then to single patients, and this can and in these patients you are measuring safety. You're documenting safety, so you always need a acceptable safety database of usually a few hundred patients. So this is in this case, if the effect is not immediately affecting as an individual patient, but just the population, you still have the safety aspect measured in the individual patient. And I mean, it's really essential that such a drug would be extremely safe, because nobody would accept a drug that has really safety concerns, doesn't really benefit the individual patient, but potentially the population. So that would be for sure not be acceptable.
So, okay, so let me ask for a different question. So going back to the microbiome, what do you think is the role of probiotics, lactobacillus and bifidobacteria in antimicrobial resistance? And this is coming from Paul Manish.
Yeah, I mean, these are long known concepts. Since decades we are using these probiotics, but it's very rare to show really a benefit in randomized controlled studies. So if you want to show an effect against some resistant bacteria in the microbiome, it of course gets almost, I don't know how you would do that. It's much too general. So at least you would need to engineer these probiotics to do something specifically. So just taking probiotics and hope that you have less resistance problems, that's not realistic. So again, it depends on really a detail. What are these probiotics doing? Are they engineered for a very specific effect? Or is it just a general approach? I'm not quite sure if the question was that general.
So Laura, yeah, Laura,
perhaps I could
please
add
go ahead.
something.
Yeah.
So the bacterial species the questioner has indicated are those that are commonly found in probiotic drinks that can be purchased now over the counter and don't tend to be used in treatment. The indication where I'm familiar with it might be given to patients and has been is when patients have had a chronic diarrhea and you're looking to help re-establish the microbiome, but that's not a treatment for a bacterial infection. So I think that we have to be very clear that the uses of that type of approach versus treating a bacterial infection. So the probiotics we have at the moment tend to be used for symptom relief and to help establish the normal microbiome after another event. Now, in terms of what else could there be for general public health benefit, there have been suggestions of adding components to these probiotic type drinks that might displace drug-resistant bacteria or even displace resistance determinants within those bacteria. But those studies and those approaches are very much in early days, and there needs to be a lot of work done, in particular with animal models, before we can even think of going forward into the general population.
Thank you. Here is something which is dear to GARDP's heart. So there's a question here, and perhaps Laura, you can start on this. It's from Emily Mayhew. It's asking about what is would these research strategies in non-traditional antibiotics, would they be delivered, how would they be delivered to low and medium increase countries where the income is lower? Would it be possible?
Well, I think the first thing is which of these approaches are going to be effective anywhere in the world. And as Ursula has clearly indicated, for many of these approaches, being able to manufacture and distribute will be very important. And that will in turn affect the cost of the product, if that's what we would be calling this at that stage, and whether it would be affordable. So it's the same question as for any approach, whether it's traditional or conventional, a non-traditional or conventional approach. So it's how much does it cost to make the product? Can you distribute it in those regions? So if it was a probiotic and it was a yogurt such as many people are familiar with at the moment, clearly there's an issue then with refrigeration, or whether you could then supply it as a freeze-dried type product. So it's going to be on a case-by-case basis, and there's no reason why you couldn't ultimately distribute these globally, but it depends on the product and how it's going to be manufactured, distributed and stored.
There is a question here from Frank Otto Gombert. It's an interesting one. Where do you see the organ, infection site, or bacterial species benefit of non-traditional approaches? Perhaps both of you can address it, Laura and Ursula.
Yeah, this is a little bit general. This organ specificity is usually achieved by some kind of vehicle that's really targeted towards a specific organ. So in general, if a therapeutic is applied systemically, then it's quite difficult to target it to organs if it's not an elimination organ or something. So I'm not sure if that's possible without using some very targeted specific approach for carrying a drug to something specific. So far, I'm not aware of any strategy, provided that you want to apply it systemically. But of course, you can you can have topical applications. You can apply something during a surgical procedure, like in joint surgery. You can apply something during the surgery. And that would then really be very specific for this joint. You can inhale something, then it would go into the lung. Otherwise, I'm not sure if there are technologies out there that are really researched so that it can be turned into a therapeutic.
Laura, perhaps I can put this same question in a little bit of a different manner and say, you know, of all these non-traditional approaches, which do you think offers the most promise?
So this depends on the purpose, really, whether realistically we're trying to prevent an infection or treat an infection. And the specifics, again, are very important here. So if you think about the fecal microbiome transplants, they are very effective. Some people are now quoting figures of about 96% cure rate. Some antibiotics could only dream of having that level of cure rate. But of course, this is specific for C. difficile. So as Ursula said, you know, it's very clear that for the fecal microbiome transplants to be reduced to what are the active components, that's very challenging. But should that be done, and of course we have to recognize it may not just be the bacterial components in that transplants that are effective, but if that could be done, that would be very useful. And so I think that's a very promising approach. I think also that the science of working with phages is advancing very quickly. But of course, at the moment, we do have that issue that this is patient specific, so it is personalized medicine. And this is not likely to be a treatment that's off the shelf for anyone at the moment. So although these are promising approaches, there are challenges to them.
Thank you. If you were to think about the use of these various non-traditional approaches, would you consider them to be used in combination, either phages, lysins, or any of the other approaches with antibiotics? Would that be possible? And either of you can take that.
Yeah, I think that will be the future, especially for more severe infections. I think it would not be acceptable to use something new where you don't even know if it really works to develop without an active antibiotic. So it's essential that you are also studying that in combination with antibiotics, and hopefully there are synergies and not any safety concerns. But this is what we call adjunctive therapy, and I think that that's the only way of clinically develop this approach, with of course all the problems of showing superiority. I mean, you have to show that it's better than the antibiotic therapy alone, and that requires a really careful selection of the patient and the indication.
So if you follow up on that, could you then say what are the opportunities for selecting these specific niche indications for the development of non-traditional therapies?
Yeah, so because these therapies are usually focused on more on patients, and it's going into the direction of patient-specific therapies, the indications are usually also niche indications, because you have to be able to show that it's better than traditional therapy, and that's very difficult to show in indications like urinary tract infections. So niche indications are really the way to go. The only problem is they have to be frequent enough to find patients for the trials, and they have to be frequent enough in one hospital, because it's not enough to have, let's say, 10,000 patients in the US. If all these patients are really distributed in all over the country, and you have one patient per year in one hospital, then you cannot do a clinical trial. So having the best is that it's an indication, a niche indication that is concentrated in specific hospitals or departments. Yeah, then you have to find an indication where there is a clear need for clinical improvement, and where a substantial and medically relevant benefit can be expected. Then a niche indication is fine, and maybe even the way to go.
So to follow up on that question, what are your thoughts on therapeutic antibodies for narrow spectrum bacterial infections?
Yeah, antibodies are usually targeted towards a specific species. So we have antibodies, usually it's Pseudomonas or Staph. These are the most common ones. Yeah, you have to show again that that it is a benefit to the patient, because it's also given as adjunctive therapy or as prevention. And that means you have to find the patients where you expect this pathogen to be frequent enough. And that's the main challenge with antibodies against a single pathogen. So for instance, if it's Pseudomonas, then you have to find the indication and the situation where you have a high frequency of Pseudomonas. But that's how usually antibodies work against the single species.
So to go back
so if I could just add,
yeah, Laura, go ahead.
Add something to what Ursula has just said, it's important to note that some infections are chronic, they're not acute, and if you're trying to treat a chronic infection where you know your patient is infected with, say, Pseudomonas or Staph aureus, then that is the more likely use of these types of antibodies in the short term. For a patient who presents in a hospital acutely ill, needing good treatment when you have not got diagnostics to tell you what is causing the infection, then you can't use these narrow spectrum treatments.
Laura, could you follow up on that and talk about in the chronic situations, especially with the anti-virulence approaches?
Yeah, so anti-virulence approaches such as for that being developed against Pseudomonas infections, such as against the type 3 secretion pathway, one could imagine that those could have a use in patients who are chronically infected, such as those with chronic obstructive airways disease or with cystic fibrosis, because they typically carry these bacteria for months, if not years. However, that does have to be tempered, that enthusiasm, by the fact that there is now evidence being produced to show that once infections in these patients are established, i.e. become chronic, that some virulence factors can then be turned off. So it has to be very clear that the virulence factor that is being targeted is still produced by the bacteria in that patient's lungs, for instance. So that's an additional hurdle for the approaches.
Thank you. Thank you. Here's a question on phage therapy and its effectiveness against intracellular bacteria. Ursula, would you like to take that?
Yeah, I mean, this is an important question. And I'm not sure if I'm the right person to respond to that. I think that there have been studies done, but there's no approach yet that goes into this question. It is difficult enough to research even the systemic distribution in the body and what happens with the phage in the body. So then going into this bacteria, of course, phages, I mean, are designed to inject or to kill staphylococci or intracellular bacteria in general. But if they have to first go into cells, I don't think, or at least I'm not aware of any studies with phages to address intracellular, it's usually staphylococcal infections.
Laura, do you have any thoughts on that? Do you have any, know
Yes.
anything about that?
Yeah, so my immediate thought was salmonella. So, for instance, we know that that's an intracellular infection. So the phage would have to get into the host cell where the salmonella is residing. And I think that could be a substantial challenge. And I've not seen any work showing that phages can effectively get into eukaryotic cells and then kill the salmonella within. But it would be very interesting to see if anyone is currently doing that work and what outcomes have been. In terms of using phages against intracellular bacteria when they are not within a person, then clearly there's a lot of activity looking at their use in preventing infections, food security, animal health as well. But that's sort of away from a treatment in people.
We do know that phages are being used to deliver CRISPR type therapies to eukaryotic cells. So maybe someday it would be possible. So we'll wait to see that.
Yes, engineered phages
Mm-hmm.
so that they target the right set human cells, which have got the target pathogen within, of course.
So let's move a little bit to regulatory because we've discussed a lot about that. So we've talked about PKPD and Ursula, you had mentioned that it's now not possible or difficult to do PKPD. So dosage, how would one determine the dosage for these therapeutics? And perhaps Laura, you could also address those together with Ursula.
Yeah, this is really a major challenge. In traditional antibiotics, we have now very well established models, PKPD models, for dose selection, and we can predict clinical outcome with these preclinical models. If there is a drug, for instance, anti-virulence drug, that does not have an MIC to measure the pharmacodynamic part of PKPD, then you cannot use the established models. Anti-virulence drugs don't kill or don't inhibit, and that's usually measured with MICs. So you are missing one part of the equation, and that at the moment makes it really impossible to use these established models. It does not say that you cannot develop new models that would be very helpful for dose prediction, but it needs to be very new, and we have not really developed these new models yet. It could be a model based on specific biomarkers, but they need to be really quantitative, and you would need to be able to correlate them with concentration and dose. And then they have to be validated in clinical studies. So we need new methods that are validated in clinical trials, but we don't have them yet. I know that there are some places where they try to work on these new models, but currently we have mainly try and error strategies, very similar to the early days of antibiotics, where we also just used any dose, whatever, without predictive models, and some were also not really appropriate.
So there is a question on a model, so let's talk about that. What's your opinion on the use of Galleria melonella as infection model in the development of anti-virulence agents?
Yeah, I just know that this organism is used really increasingly for all different kinds of questions, and also for antivirulence approaches. But in detail, I'm not sure if we have already a solution, or if this will be a useful model for the future. And especially because we don't have a correlation to clinical outcome. So that's all what I can say to this model.
Laura, any thoughts on models for testing these systems?
Yeah, so yes, the Galleria model is widely used. It has a very easy readout in that the larvae usually go black when you can read this out when it's dying or when it's surviving. It's cheap, and you don't have the issues that some countries have in licensing researchers to do experiments with animals such as mice. So it's a very useful step. It minimizes using animals until you have a candidate you really wish to go forward with. But it is still accepted that Galleria does not replace doing experiments in mice, and there is no alternative at the moment, and that's before you even go as far as doing anything in humans.
All right, thank you. Do these new drugs have any priority with the FDA? Who would like to take that, Laura or Ursula?
Yeah, I know that there are lots of discussions with FDA and EMA, and there have been several workshops discussing all these issues, and FDA and also EMA are very interested in helping to solve some of the regulatory issues. And we could see that with phage, especially CMC, and also with establishing the microbiome libraries and repositories. So there's a lot of interest and helpful attitudes, but there's no priority. There's no priority, and they see what's coming, and if there's a new approach that can convincingly show that it's a benefit to patients and it has no specific toxicity and can produce reproducibly, then I think they are open, but there's no priority.
Do you have any insight from the UK regulators there, Laura, or anybody else?
Well, the UK is, as you know, under EMA as well. So that would be the first, well, until the 31st of January, that's just gone, of course. But at the moment, nothing is changed there. And the reality is for any new treatment, efficacy is all important. And so that has to be shown, and you have to have a safe product. So there's nothing substantially different in the regulatory pathway to that for a conventional drug. But it's the endpoints which we've just been discussing a minute ago. How do you measure that endpoint?
Very good. To continue on the type of molecules going forward, there's a question on the conjugates. So if you take an antibody conjugated to an antibiotic, would that be useful, and how successful could those be in development? Because you'd have both the antibiotic as well as the antibody treatment for, let's say, Pseudomonas.
Yeah, I mean, we have one example in clinical development, and this is now targeted to intracellular stuff, infections. The indication, at least for the clinical study, was recurrent bacteremia caused by Staph aureus. Yeah, I mean, this is again a niche infection, and it's really difficult to show an effect if you can cure recurrent bacteremia or can reduce recurrences. It's a question of how to show a benefit of this new drug. It always comes back to how to show a benefit.
That's right. Okay. You did not address antimicrobial peptides. Would you like to comment on those, either of you?
I mean, as long as they are direct acting and not just immunomodulating approaches. So if they are direct acting, then you can look at them as all our other antibiotics, at least from the clinical development. There wouldn't be a lot of difference.
Okay. Here's a general question as we're getting to the end. I'll pursue this one. As we all know, there has been investment issues for antibiotics. And so there's a question here on, is there any difference in the interest of investors in non-traditional antibiotic therapies versus antibiotics? So for either of you, if you can take that.
Yeah, I'm starting with this. It is really clear for microbiome-modulating therapies, there's a lot of interest from investors, but it's not really so focused on antibacterial therapies. It's more on broader ideas of curing everything from Alzheimer to multiple sclerosis to whatever, obesity. So they see just a huge promise behind the entire technology. And sometimes I have the impression that C. difficile is just used to somehow to start the technology and get experience. Yeah, so there's a lot of interest. For phages, there was a lot of interest of investors, but some recent failures and problems of establishing therapies beyond passionate use. I think the enthusiasm by investors is not that great anymore.
Thank you. I think we are coming to, Laura, would you like to say anything about that? Or should I go to the next one?
So I'm happy to comment about investment. I think we're all aware that investment for research and development of any new antimicrobial, whether it's traditional or non-traditional, is extremely challenging at the moment. And investors seem to need even more convincing that this is an area that they should invest in, because they can then anticipate a reward of their investment. And that is the challenge. So I don't think there's any difference between what type of antimicrobial it is. It's just there really isn't a huge amount of investment specific to the antimicrobial area. As Ursula's just mentioned, microbiome investment is a different thing, and antimicrobial aspects of microbiome treatments are sort of following on, they're sort of on the coattails, as it were, of this area.
So I want to ask one question myself, which I think is important for all proteins and peptides being used. If they're going to be used in repeated chronic therapy, what is being done with an immune response, or can they only be used once? Laura?
Well, again, it depends on the specific type of protein or peptide, and if they elicit an immune response quickly and after first exposure, then it may be challenging to use repeatedly. But it really does depend on the specifics.
Yeah, may I just add, a typical example would be endolysins. At the moment, there's really an issue with immune response, and that would limit the dosing regimens to maybe a single dose. But that could be also overcome by some kind of engineering. So also this, despite this problem is known, it may be possible to overcome it.
Thank you. Thank you. So with the time constraint now, so we have now come to the end of our Q&A session. There are some more questions, but I think we have pretty much answered most of them or covered it in the talk. I would like to thank everybody for their questions and comments. And once more, thank you to Ursula and Laura and to Astrid for setting up this very timely webinar. Astrid, I'll pass it on to you now.
Thank you very much, Prabha. Thanks for moderating this webinar. And of course, thank you, Ursula, for presenting and Laura for contributing to the discussion. I would now like to announce our next webinars. On the 27th of February, we will have Alexander Lepak from the University of Wisconsin, who will talk about PK/PD murine infection models. And on the 26th of March, Shampa Das from the University of Liverpool will present in a webinar on the topic of PK/PD target attainment analysis for dose selection. You can already register for the February webinar on Revive, and we will also open the registration for the March webinar very soon. Now, one final comment. We are currently running a survey, which I would like to bring to your attention. We are considering to ask speakers of our previous webinars, mainly our 2019 webinars, if they would agree to join us for another live Q&A session. But of course, we will only do so if there is sufficient demand. So therefore, I would ask you to please fill in this survey and let us know for which webinars you would appreciate another live Q&A session. The link for this survey will be shared in the webinar follow-up email, which will go out the next week or the week after, as well as on Revive. And with this, I would like to thank everybody for joining today and for contributing to the discussion. I hope, as always, that you found it interesting and useful, and that you will join us for our future webinars. And please make sure to spread the word in your networks and encourage your colleagues to join as well. Thank you and goodbye.